It is no trouble at all to be here. In fact, it's like the deepest honor, the greatest pleasure that I have is to get out and interact with undergraduate students because I'm at Upstate Medical University. We don't have any undergraduate students. We train medical doctors in our College of Medicine. If you want to become a doctor, maybe you'll come to Upstate someday. We train nurses in our College of Nursing, if that's the path you're interested in, from bachelor's degree all the way up to doctorates in nursing. We train health professionals like occupational therapists, physical therapists in our College of Health Professions, and we train scientists. And in our College of Graduate Studies, as Dr. Peltz mentioned, we have a program in neuroscience that grants master's degrees and PhDs, but we also have programs in cell biology, anatomy, biochemistry, molecular biology, et cetera, et cetera. But I don't get to interact with a lot of undergrads. And I think you guys are at this inflection point in your lives and your careers, and to be here, to be welcomed in, and to be able to speak with you guys about my journey and about the journey you guys might like to undertake, it really feels magical. So it's my great pleasure to be here. I'm glad we could reschedule. Sorry about last week. I've had a mess of car troubles this winter, two crashes, none my fault, by the way. But it's treacherous out there, but I'm glad I made it here. So if we don't do more than one slide today, I'm totally fine with that, because I'm here in service of what interests you guys, but I've planted a seed here called research domain criteria, and I'll explain what that is, why it matters, but really it's in the context of the thread that ties us all together and what brought us here, and also the people on the recording that maybe kept you away is like feeling overwhelmed by this enormous field of psychology and then the medicalization of that in psychiatry. How do we make inroads? How do we as professionals get our own niche? How do we make a living doing this? But how do we ask questions that are answerable? Because so many of the questions seem so big. Like, what is consciousness? Or questions in sleep are, massive, not understood well. How do you start to formulate questions and hypotheses that are testable things, right? And so research domain criteria is one of those undertakings that I've engaged in that kind of helps us operationalize psychiatric disorders, particularly, in a way that is different from how clinicians treat them, okay? So, if you have any question at any time about anything I've said or anything that what I've said triggers a question for you, ask it, because I'd rather use our time on that than getting through 100 slides or something. I'll plan to talk for like maybe 50 minutes, but honestly, I mean, we'll just use the whole hour as a dialogue, I hope. And so questions, answers, slides, whatever, we'll get through as much as we can. And of course, if you have questions after the fact, you can just reach out to me as well. I'm happy to answer those questions too. So, but our starting seed is research domain criteria. So about me personally, I'm a New York native. I grew up in Utica, New York. Many of you guys have passed Utica and probably never stopped there on your way to Albany or to Vermont to go skiing or whatever. But I graduated high school in Utica, New York, and then I came out to Syracuse University. I started as a biology major, and I thought I wanted to be a marine biologist. A lot of kids in my generation thought we wanted to be marine biologists until we got to college and had to take bio classes, and it turned out to be really hard. And I literally almost failed out of college as a bio major. Like, I failed many classes. I got a D in genetics, many other failing grades. What saved me really was psychology. And I did graduate as a double major in biology and psychology. I don't know how I managed that, but psychology was much more intuitive to me. Even though it's less concrete than biology, it's much more intuitive to me. And I think that's probably what made you guys kind of think, yeah, I should probably be a psychology major. There wasn't really a neuroscience major back then that kind of blends the best of biology and psychology and embeds kind of biological thought in the brain and so on. But I managed to graduate somehow, and then I went to the very esteemed Northeastern University, which back then was not as esteemed as it is today. It was one of the few schools that I got accepted into for graduate school because of my miserable record. I had a 3.0 GPA when I graduated CUNY, I mean, that's not anything to write home about, right? But it was a blend of good and bad grades. But I had found what I wanted to do, which was research. And I ended up in a lab that studied the developmental effects of prenatal exposure to drugs. This was 1996. So it wasn't that far removed from the kind of crack baby epidemic of the 1980s. You guys may have not even heard of this. You guys are of a different generation, but there was this epidemic of crack in the United States, and then there was this epidemic of concern about the developmental effects of that in utero exposure to cocaine, particularly this concentrated form of cocaine on offspring. So I went to a lab that was studying this, and I thought, oh, this is going to be really cool. And the first week I was in the lab, I remember the senior graduate student, he showed me the types of experiments we were doing with rats and injecting them with drugs, and I was like, oh, that's kind of squeamish, but okay, you got to do that. And then, taking a live animal and cutting its head off with a guillotine so you could access its fresh brain tissue. And I was like, that's gonna be heavy. That's gonna be tricky. I am ashamed to admit I didn't have the backbone to kind of walk away from that. I was in graduate school. I wanted to progress. I wanted to do what my PI wanted of me, and I did those experiments. But I grew physically and morally allergic to animal experimentation. This is just my perspective. I didn't like cutting rats' heads off. I still think about that and think about how shameful that was for me. Everybody makes their own decisions. But I also grew physically allergic to it, which made me realize there's no way I can do this for the rest of my career. So it was not until the third year of graduate school that I figured out what I really wanted to do. And I made a list of criteria that I said, this is what I want to do. I want to study the way that genes and environments interact to make people sick. And not just sick like cancer or heart disease, but have mental disorders, have mental illness, have symptoms of psychological distress. That was the number one thing. I wanted to study humans. I didn't want to study animals. And I really wanted to hone, just as a personal kind of curiosity and habit, quantitative methods, skills in quantitative methods. I love numbers. I love formulae, right? We talked about brain genie and mapping functions. I like algorithms, right? And I always have. And so I wanted to kind of pick up this skill set in my next endeavor. So I didn't know the field that I wanted to move into at all, just that it was there. It was out there. And it was a field called psychiatric epidemiology. And epidemiology, who knows what epidemiology is? I think we have a better sense of it now after COVID, but I mean, who wants to put forth a definition of epidemiology? Yes, sir.
Is that related to epigenetics?
Not technically, not technically. Epigenetics is kind of the study of how our environments get imprinted on our genome. Epidemiology is more related to the science behind epidemics, in a sense, meaning the distribution and determinants of disease. So always remember this definition, the 3 Ds, the alliteration of it. The distribution and determinants of disease, that's epidemiology. And not just epidemiology, but psychiatric epidemiology, the distribution and determinants of psychiatric illness in populations. That's what I want to study. Let's make it even more complex. Psychiatric genetic epidemiology. What does that mean? It's the distribution and determinants of disease in population, as well as the transmission of disease vertically in families. And now if you think about what that means, are psychiatric disorders inherited? Well, think about your own families, and we'll talk about some data on this topic. Think about families you know. Lots of traits are passed through families, right? But that doesn't mean that they're genetic. You think about your own parents and what language they speak. You speak the same language as your parents generally. That's not because of any genetic or biological function. That's learning, right? That's entirely environmental. And most things in psychiatry have an environmental component to them, a learned component to them, but it also turns out that they do have a genetic component, and I can show you guys the evidence for that. So it turns out that psychiatric disorders exist over a range of subtly heritable versus very heritable, and everything in between. But most aspects of the human mind are some part nature and some part nurture. It's not an either or, right? The debate, the battle between nature and nurture is settled as far as it concerns human psychology. And it's really that it's a compromise. There's no longer a war between nature and nurture, it's a compromise. Both parts play a role. And I wanted to invest my career in figuring out how much of that risk for a psychiatric disorder is environmental and how much is genetic. When I went to, I wrote a letter to a team that was working at Harvard Medical School and the Harvard School of Public Health. They didn't answer my letter, but 6 months later I wrote to them again and I said, I really want to do this. Can you guys pay attention? And I finally got an interview and the rest was history. But when I went to talk to them about the work that they were doing in this area of psychiatric genetic epidemiology, I noticed that they recognized the importance of the environment, but they were really focused on gene hunting. They wanted to find the genes. And I told you already, I got a D in genetics. I was not keen to dive into genetics. I wanted to focus on the environment. And what they told me changed the trajectory of the rest of my career, which is that genetics is a complex science, but it's very rule-driven, and it is indeed finite. There is an end or a limit to the amount of genetic information that we have as humans. And when you change one thing in our genome, it has predictable consequences for downstream biology, whereas the environment is infinitely more complex than the genome and very hard to quantitate and control. And so if you go down that path of studying the environment, you're doomed. Pour yourself into genetics. And I did, and I taught myself genetics because their job was not to teach me genetics. I taught myself and epidemiology and so on. And the rest is really history. That was 2001. And at that time, that was 25 years ago, we did not have a single gene that we could point to as a bona fide, legitimate risk factor for a psychiatric disorder. Not one, not a single one. We knew they existed, but we could not find them. And here we are 2.5 decades later where there are literally hundreds that are known to be reliable, bona fide genetic risk factors for psychiatric disorders. And we could talk about them and their effect size and their penetrance and so on. After that, I went to UC San Diego for 3 years, and then 20 years ago, I came back home to upstate New York, where all my family is, to raise my own family here. And I'm at SUNY Upstate Medical University ever since. And I established my lab there, and I'm affiliated with the Department of Psychiatry primarily, but also neuroscience and public health. And I should put another arrow on this because I just got my master's in public health last August from Emory University, which I'm super pleased because that's the next phase of my career. Is the past 20 years I've spent gene hunting myself, trying to find genes that predispose toward mental illness. The next phase of my career is really about how do we take that information into communities, understand what puts people at risk and how to intervene to keep them from developing psychiatric illnesses. So that's how I got here. So this is my lab. This is the Psychiatric Genetic Epidemiology and Neurobiology lab. So not only do I continue to do psychiatric genetic epidemiology, that is, study the distribution and determinants of psychiatric illness in populations and in families, we also do neurobiological research, introducing genetic mutations into, for example, cell lines and finding how those mutations that are known to be associated with risk for a psychiatric disorder, how do they change biology of cells and make those cells more vulnerable to stressors? And then at the level of an organism, how does that lead to presentation of psychiatric symptomatology? So that's the PsychGene lab, essentially. You can find us and our work at www.psychgene.com, psychgenelab.com as well. And the mission of the lab is a little bit different now than it was when I started it. When I started it, the mission was, we will develop and apply methods in pursuit of the determinants of mental illness. Does anyone see the subtle difference in there? The subtle difference now is that we also are interested in mental health. Not just the determinants of mental illness, but the determinants of mental health. And correspondingly, the vision is that we'll discover determinants of mental health and mental illness and develop interventions founded on those discoveries. And this has been on the wall since 2009. And it kind of guides the people who come to the lab, the work that they want to do, where they see this work headed. But it was okay, we found, to kind of change that vision to include mental health at a point where we had been so successful, not me personally, not even my lab, but as a field, in banding together to identify the determinants of mental illness, that I could look past that part. We know a lot about what makes people sick, but what we don't understand, and what I found really captivating, and what I've turned my lab toward about 8 years ago, is how do some people inherit a ton of risk genes for a psychiatric disorder and exist and grow in a very traumatic environment and yet still seem to hold their shit together? How do people remain, yes, I swore, I'm sorry. How do people remain resilient in the face of both inherited and exposed risks? And that to me is really compelling, because once you start to see, okay, we understand if people have this level of risk, they're much more likely to develop psychiatric illness. What's really compelling to me as someone who wants to find ways to intervene is not studying those people who've gotten sick, but those people who've been able to stay well. And so then we transitioned our focus really toward resilience full time. And it's what we're no longer really in the business of kind of gene hunting for risk genes. We're looking for resilience genes, things that people have inherited that also protect them from the other bad things they've inherited or the exposures that they've had that in a typical person might lead to an increased liability toward illness. So that's really compelling, and we changed to adapt to that. Any questions so far? Okay. So this is what the inside of my work brain looks like, okay? At the center of it all is prevention. And you guys can probably get the sense, like, I'm a very visual guy. I'm very schematically oriented. I love acronyms. I love visual points of light, as they say. This, to me, helps me make decisions about the types of work we want to do. And I admit that I'm in the most privileged position on this earth, in that I get to teach, I get to serve my university, but mostly what I get to do is research. And of that research that I do, I get to choose what I focus on. And that is such a privilege, and I never take it for granted. But in exchange for that privilege, I want to make a difference in the outcomes that we experience as a society by decreasing the incidence, the prevalence, the morbidity, the mortality associated with psychiatric illnesses. And that's why I went back to school to get my public health degree. Because at the center of it all, I really want to see us be in a position where we're preventing cases of schizophrenia, where we're preventing people from having to develop bipolar disorder before we treat them, right? By understanding their predisposition and intervening prophylactically so they don't have to develop psychosis, or that a kid doesn't take their suicidal thoughts towards suicidal attempts. I think prevention's where it's at. But preventing things is really hard. It's not simple. And that's why there's this big wall around it. It's kind of, an insulated goal. By looking for risk factors, this is the gene hunting that we've been doing all along, and trying to identify biomarkers, which is a separate talk, you can't really make inroads on prevention. Like, just understanding the genes that someone has inherited that put them at risk for a disorder, what do you do with that? Not much. You can say you're at risk, but what do? You have to understand the biology that is engendered by these specific genetic mutations so that you can intervene at the biological level to prevent that, that cascade towards symptomatology. And the same thing with biomarkers. I've dedicated actually a lot of my professional life toward trying to find biomarkers for psychiatric disorders. And I think it's been a valuable teaching because we do need biomarkers in psychiatry, and we don't have good biomarkers in psychiatry. Why does that matter? Well, we have very many well-trained clinicians who you can go to or bring your child to or family member when they're in psychological distress and say, diagnose my kid, diagnose me. And they can do that quite reliably, and that's what we'll talk about is the reliability versus the validity of those diagnoses. They can do that quite reliably, but there's not a guide that attaches those reliable diagnoses to anything biological or objective. Even though we can train people to be reliable at this, there's still an element of subjectivity to it. And maybe we should have brought a version of the DSM or a few versions of them, because the DSM, which if you don't know is the Diagnostic and Statistical Manual of Mental Disorders of the American Psychiatric Association, it's the set of criteria, big set of criteria, by which these psychiatric disorders are diagnosed. And in other countries, they mostly use what's called the International Classification of Disease, but it's very similar. It's a set of criteria with checklists that say if you have these symptoms, 6 out of 9 of these symptoms, you have ADHD, okay, attention deficit hyperactivity disorder. So, but there's an element of subjectivity to it. It needs a well-trained clinician, and it needs insight from the person you're diagnosing. They have to be able to be a reliable reporter of their internal world. But the diagnostic criteria for psychiatry change about every decade or so. New disorders are added. Some disorders are no longer considered to be disorders. You'd be aghast at the list of disorders that is in DSM-3 at this point. So how could, the criteria for whether or not you've broken your leg have not changed in the past 50 years or 100 years or even 1,000 years. When you're playing basketball and you jump and you fall awkwardly, you hurt your leg and you can't walk, you might know right away, oh, I broke it. Or you might have heard something. But it could have been a ligament or a tendon. Go to the hospital, get an x-ray. When you get that x-ray, it's literally black and white, and it's clear and objective. You've broken your leg, or you've not. And that suggests a course of treatment. In psychiatry, we have nothing like that. And that lack of objectivity, that lack of rapid result, I think undermines confidence in psychiatry. And I think that's why, in America and other places, but particularly in America, I don't want to get on a woke rant. I don't want to. But there is an anti-psychiatry movement in America that has predated the anti-science movement ushered in by Trump. That's all I will say. The anti-psychiatry movement, in large part, was born right at Upstate Medical University by a guy named Tom Zaz, who was a psychiatrist himself, whose views on psychiatric disorders changed over time. He took a kind of counter-psychiatry position that was probably too extreme, but to stake out a position toward personal autonomy and against the medicalization, particularly against the forced treatment of people with psychiatric illnesses. I think we've landed in a kind of middle ground, but it just goes to show that we don't have those objective, laboratory-based biological criteria for psychiatric disorders that they do have in other branches of medicine. And that leaves us open to doubt and to stigmatization. People won't get treatment as early because they're afraid of the stigma of psychiatric disorders and so on. So I feel, this is all to say, I do feel we need biomarkers of psychiatric disorders desperately. And I think we've made good inroads toward that. But again, getting a biomarker test meets one goal, but it doesn't meet my ultimate goal. It doesn't help me prevent anything. In order to prevent something, you need to understand the mechanism up at the top there. You need to understand how those biomarkers got disturbed, or how those risk factors disturb biological processes to change your brain to make it susceptible to showing symptoms. And so that's why my lab does neurobiological research as well as epidemiologic and biomarker research, is to try and find the biological mechanisms by which inheriting certain mutations changes your neurons, changes your glia, changes your brain circuitry to make you more likely to express these particular symptoms. And the bottom part, is another challenge in psychiatry in that there are 1,000 different ways that brains can go wrong. There's a couple different ways you could break your leg. You could have a compound fracture, a simple fracture, dislocation. But there's 1,000 ways to, quote, break a brain to make it experience psychiatric disorders. And few of those ways are as clean as a broken leg. Mostly, people, comorbidity, as we say, is the rule. It's not the exception in psychiatry. And so if somebody has depressive symptoms, they may also have anxious symptoms. If they have attentional symptoms, they may also have a higher likelihood of anxiety, and so on and so forth. So these are what we call phenotypes. Phenotypes are like the listing of disorders in the DSM. Those would be considered phenotypes because they're describable phenomena that could ostensibly be mapped onto genetic, biological, and environmental exposures, right? But we change the definitions all the time. It's like building a bus while it's rolling down the highway, kind of. We're studying these things. Autism is a great example. Autism spectrum disorder used to be a category that comprised 5 different diagnoses, like pervasive developmental disorder, not otherwise specified. Asperger's syndrome is not in the DSM. It's gone. It doesn't exist anymore, right? Tell that to somebody with Asperger's syndrome. But now they're all lumped under one diagnosis, which is autism spectrum disorder. What does that do? Well, when you used to study autistic disorder from a genetic point of view, there were strict criteria. It was a pretty severe and significant phenotype, far less severe than Asperger's syndrome or some of the other autism spectrum disorders. And so it might be more rare, but it was much more homogeneous. And that made it kind of easier to study. When you say, well, let's study autism spectrum disorder instead, you're kind of watering down any genetic signal that was there to be found. And so these changes in the way we define the things we're trying to study have big implications because what genes you can map onto a phenotype or what biomarkers you can find for a phenotype depend on how you define that phenotype. Okay, so that's why this is kind of hashed out here. And the way you define those phenotypes, it leads to kind of an atherosclerosis, like a thickening of the arterial wall of flow of information from the phenotype to the risk factors and the biomarkers. So what we're really focused on today is the top, which is mechanisms, and the bottom, which is the phenotypes. And that's where we circle back to research domain criteria. Are we doing okay so far? I hope I haven't alienated people by getting on my political soapbox, but if so, so be it. I accept that. And then in my lab, having this model kind of guide the way I do my work, then I just populate it with technologies and disorders and things I want to study. Okay? So let's talk about what we do know about psychiatric disorders as they're defined in a particular version of the DSM, let's say DSM-5. What do we know about these psychiatric disorders as a genetic epidemiologist, as somebody who wants to understand mechanism, but first has to identify genetic risk factors? Because I'm not getting into the environment. I just don't want to deal with it. Okay, well, do these things run in families? How do we establish that? Has anybody ever seen a pedigree drawing like this before? It seems kind of familiar to people. Does anybody understand the symbols and their relationships? Yes. Yeah, so you've seen this before. So the squares represent men, the circles represent women, and the levels of the tree represent generations, right? So on the left-hand side here, we have a pedigree, which is a family drawing, of a case. That means somebody with a disorder of interest. Let's say schizophrenia. That's the first thing I've studied, and it's always stuck with me. And the proband is the person here with the arrow that brought this family to our attention. Maybe they're a patient in our clinic, or maybe they're the person who responded to our ad. They meet the criteria for schizophrenia, and thus, I want to study your family, okay? Now, we can find, so let's say this person is affected with schizophrenia. We might color that in or indicate that in some way. We want to know how many other people in their family also have schizophrenia or a schizophrenia spectrum illness. We can find that out 2 ways. One of them is a cheap and easy way, which is to ask the proband, did your mom have schizophrenia? Does your dad have schizophrenia? Do your grandparents have schizophrenia? That's cheap, easy, but only fairly accurate. And the more severe the phenotype is, the less reliable the reporting is. In some instances, people will under-recognize or under-report illness in their own family. In some, they will over-report. They'll see it everywhere. I have it, oh, I see it everywhere in my family, right? So family reporting is not necessarily the most accurate, but it is cheap. You only have to talk to one person. On the other hand, you could do family interviews, which is, okay, don't tell me about your mom and your dad, just tell me how to get in touch with them. And then I'll go talk to them, and I'll do the same interview with them to diagnose them as I did with you. That's a lot more involved. The bigger the families get, the more, especially in the US, huge country, everybody leaves home. In other places where I've done work like this, like Taiwan, it's an island nation, and families stick together. And when one person in the family gets sick, like the kid, the whole family stays together and takes care of the kid. So that's easy. In the US, it's really hard to do family interviews. But you could do that. And then you find out, okay, mom didn't have any schizophrenia, dad didn't have any schizophrenia. Mom's sister, so that would be this proband's aunt, she might have a schizophrenia spectrum disorder. Do we count that? We're doing a family study of schizophrenia. Do we want to count that? Do we not want to count that? It depends. It depends what your interest is. But their mother, which is the proband's grandmother, well, she definitely meets the criteria for schizophrenia. So these are issues you could think about, but essentially, we're finding probands with a phenotype we're interested in, we're collecting their families, and we're finding out how many of their other family members are affected. And then you do the same thing for an individual here who's called a control proband, who is matched in every possible way to this case proband, same sex, same age, same geography, et cetera, but they don't have the diagnosis of interest. So they're a very well-matched epidemiologic control. And then we do the same thing, we interview their family. And lo and behold, if you are biologically related, these have to be biological ties, by the way. Adopted families aren't useful for this. They're useful in another way, though. Maybe we'll talk about it, maybe not. But anyways, these are biological family members. And it turns out that if you ascertain lots and lots of families of case probands and control probands, for each and every psychiatric disorder that you want to study, there's a higher rate of illness in the families of case probands than there is in the families of control probands. Makes sense. But just because something makes sense doesn't mean we can take it for fact. We have to prove it. And generations of scientists have proven this. And we can calculate an odds ratio from that. But essentially what you see is a correlation with genetic distance. And, well, let me show you actual data here. Those are the predicted data. Actually, these are the predicted data on the right. These are the actual data on the left. The population prevalence of schizophrenia is equal to the risk of schizophrenia in the general population, given that you don't have a known biological relationship of somebody with the illness. And that's less than 1%. It's often quoted at 1%, but it's actually less than 1%. Whereas if you have a second-degree relationship to somebody who has schizophrenia, your risk is anywhere from 2 to 4 times higher, just by being a second-degree relative. If you're a first-degree relative, like the parent of someone with schizophrenia, or the child of an individual with schizophrenia, or the sibling of someone, you share 50% of your genes in common with that person, on average, and your risk is anywhere from 6 to 14 times higher. Now look at these individuals. A monozygotic twin, which is an identical twin. Any twins in here? Identical? Fraternal. Okay. So you're in this category here, a DZ twin, dizygotic twin. That means 2 eggs, 2 sperm in the uterus at the same time. So they share an intrauterine environment, but they're only as genetically similar as any other brother and sister. Very fascinating. Whereas monozygotic twin is 1 sperm, 1 egg that then divides immediately, and they grow in parallel. They have 100% of their genes in common. And what you see is there's a big difference between dizygotic twins and monozygotic twins. Even though dizygotic twins share an intrauterine environment, they grew up together, same age, everything, but monozygotic twins who have twice as many genes in common have more than 3 times the level of shared risk. And in fact, if your twin has schizophrenia, if they're a monozygotic twin, you have about a 50% chance of having it yourself. It's not absolute, though. What does that tell us? If schizophrenia were a genetic disease, if it were caused by genes, then if your sibling has it, you should have it 100% of the time, monozygotic twin. If your monozygotic twin has it, you should also have it 100% of the time because it's only genes that are causing it, right? And what this tells us is that even if your monozygotic twin, with whom you share 100% of your genes, has schizophrenia, your risk is still only 1 in 2. That means there's room for the environment to operate. It's not all genetic. These are not genetic diseases, but it suggests that there's something genetic going on. Okay? Doesn't prove it, by the way, because I've already mentioned there are things that run in families and even things that show patterns like this that are not genetic. Things like language, religion, politics sometimes, but also tuberculosis. Tuberculosis is a virus. It's an infection, but it shows this same pattern. Why? Because it's a proxy for proximity. You live with people who have tuberculosis, and you're likely to get tuberculosis. It's not because you guys share genes. It's because you share an environment. But family data like this do give us a sense that the search for genes is warranted. Okay? I'm going to skip that. So every psychiatric disorder that I've ever looked at, yes, sir.
The 2 slides ago, what was the second child notated next to the monozygotic twin?
This child 1?
The child 2.
If you are a child of both parents who have the illness, right? So your mom and your dad have schizophrenia, your risk is like 40%, right? Whereas if only one of your parents has it, your risk is like 12%. Okay, so good question. Yes, sir.
So if someone, for example, their grandparent had schizophrenia and they themselves don't show any sort of symptoms for schizophrenia, but then they take, for example, some sort of substance, and they have substance-induced schizophrenia, would they be classified as someone with schizophrenia or not?
It's a good question. First of all, I would say if they've taken, for example, marijuana, methamphetamine, or something like that, that can precipitate psychosis, we wouldn't necessarily call that schizophrenia. And if it goes away when they stop taking the drug, it's definitely not schizophrenia. But you could say it's psychosis. And the difference is psychosis is kind of the hallmarks of psychosis are a set of symptoms called delusions and hallucinations. Delusions being false beliefs, like the belief that your thoughts are being broadcast out into the classroom, or that somebody's trying to get you, paranoid delusion, or the belief that you are an important historical figure reincarnated, a grandiose delusion. These are false beliefs. Hallucinations, on the other hand, being having a perception without a having a sensation without a sensory stimulus, right? So seeing a dragon in the corner of the classroom, hearing voices, those are hallucinations. So those are the 2 hallmarks of psychosis. Those can be very well mimicked by drugs of abuse. But if those symptoms go away when the person stops the drugs, it's not schizophrenia. If they persist, then what's likely happened in that case is that the person in question has inherited risk genes for that illness, and they've now exposed themselves to a biologically traumatic stressor, such as drugs, that has taken, in my view, from what I know about the genetic architecture of these disorders, have taken a person who's maybe at subthreshold risk for expressing the psychiatric disorder in the absence of trauma and pushed it over the threshold of risk factors needed for the system to break down. In other words, the genes, we all have risk genes for psychiatric disorders. None of us has an absence of risk genes because there's so many of them. Probabilistically, we are all carrying risk genes for psychiatric disorders. The number that we're carrying depends on our ancestry, of course, and can be determined with a very simple, very inexpensive test. The problem is it's not determinative, right? So that's why we can't use it to make accurate predictions about who will get sick. But this person, in this case, or people like this, they may have a lot of risk genes for schizophrenia because of their family history, but not enough that the genes themselves were, in the absence of major kind of environmental stressors, enough to bring out symptoms of psychosis. And those genes probably built that person's brain in a way, we might actually see, where they've had excess pruning of synapses such that they don't have the kind of reserve that a brain without such risk genes might have, so that when a stressor is encountered, like drugs of abuse, or going off to college, or moving out on your own, or opposite sex relations, or other things like that, the stressors of adulthood, then that becomes a time where things like schizophrenia and bipolar disorder, which are marked by psychosis, often come to light because there's just not that biological reserve. The genes for those disorders have made the brain such that it's operating at the bare minimum number of synapses to perform its duties, but under stress, it starts to show weaknesses. So in that case, I would say it depends. If the symptoms go away after cessation of drugs, they don't have schizophrenia, they don't have bipolar disorder, they don't have any biologically based brain disorder, they just have drug abuse. And drug abuse mimics psychosis, or produces psychosis and mimics these other disorders. But if they were to develop a long-lasting psychosis that persisted beyond cessation of the drugs, it could be schizophrenia, it could be bipolar disorder with psychosis, but it's probably because they had a lot of risk genes that they were able to keep in check, but then doing something with their dopamine system and other neurotransmitter systems by doing drugs to the point where it elicited a psychotic episode has kind of quote unquote changed the biology of the brain so that it's now got a schizophrenia phenotype. Does that make sense? Okay. Okay. Yes?
Would that also be like fair to assume that applies to like antidepressant induced psychosis or maintenance mania? So that if it were to use it seized and then the mood, I guess mood symptoms like maybe were still present after the substance.
Yeah, I mean, some of those things will actually take longer than a week or 2 weeks to resolve, but they should resolve if they were elicited only by the antidepressant. They should resolve over time. If they never return to baseline, then there was probably like latent bipolar disorder or mania there. A lot of times, bipolar disorder, for people who maybe are not familiar with this, manic depressive illness is marked by periods of deep depression and high highs, high physical activity, racing thoughts, inability to control impulses, et cetera. So, depression, depressive incidence is a criterion for bipolar disorder. It has to be there. But the manic episode may never have manifested. So somebody switching from a depression diagnosis to bipolar disorder diagnosis is quite common. That can happen. I don't know pharmacotherapy enough to say this is common for antidepressants, but I would say that probably it may have elicited a manic kind of symptomatology that unmasked bipolar disorder risk that was probably there to begin with, that never had the sufficient stimulation to bring it about. Okay. So what do we know so far? Well, we know that every psychiatric disorder as defined in the DSM has some familial basis to it, and that's what this table here shows, is that every disorder I've ever looked at, the relative risk is greater than 1. That means being the same-sex biological sibling of somebody who has this diagnosis means that your risk of that diagnosis is at least twice, and in some cases as much as 10 or 20% for things like autism, schizophrenia, bipolar disorder, et cetera. So the range is very broad, but there's no psychiatric disorder that, being biologically related to someone with it means no level of risk for you. Having a biological relationship to somebody with a psychiatric disorder always increases your risk for that psychiatric disorder. Interestingly, and this is where the DSM starts to break down, it also increases your risk for other psychiatric disorders. These things do not breed true. And thus, that leads us and has led us for decades, really, to presume that the risk for these disorders are not conferred by a single factor. They're conferred by lots of factors that coexist within individuals. Now, we've talked about the limitations of family studies. Things run in families for non-genetic reasons, but it gives us a basis to dive in. We've talked a little bit about twins as well. This is twins in pedigree notation, but I just want to return to it briefly because this was the crux of understanding the degree to which we could expect to find genes for psychiatric disorders. And again, I'm not talking about, and we'll reinforce this again and again, I'm not talking about psychiatric disorders as genetic disorders, okay? If you know Huntington's disease is another nervous system disorder. It's a neurological disorder that's characterized by shaking and loss of motor control. It's not a psychiatric disorder, although it can have some psychiatric sequelae, as they're called. Huntington's disease is a genetic disorder. It's caused by one single gene. And if you inherit a copy of this gene, where the certain sequence of nucleotides in your DNA grows to a certain length, you will get Huntington's disease. There is no stopping it. There is no denying it. If you get this sequence of a certain length, I think it's like 18 trinucleotide repeats or something, you will get Huntington's disease with 100% certainty. And if you get Huntington's disease, it's because of this. In that event, it's a genetic disease where the genetic mutation in the Huntington gene is both necessary and sufficient. And this is a phrase you should get accustomed to hearing, okay? It's necessary because you can't have Huntington's disease without it. You might have some other neurological disorder, but it's not Huntington's disease unless you have this. And it's sufficient because it doesn't matter what your environment is. If you have it, you're getting Huntington's disease. Necessary and sufficient, yes.
Maybe a little off. Why is it like the 2-week mark? Like after 2 weeks, if you're continuously like feeling this way, then and like you answered the questions, however, like 1 to 4, why is it like the 2 week?
Well, maybe Dr. Peltz knows this better than me. He's a clinical psychologist by training, but I mean, the stability of these things is really important. And just like, talking about the incidence of somebody whose psychosis might be attributable to some passing phenomenon, such as drug exposure, so too, depression might be symptomatic of a job loss, a loss of a spouse or family member. So the persistence of these things, regardless of the environment you find yourself in, makes the difference between something like grief and true depression.
Yeah, I mean, the fact that we go through periods of happiness and sadness, feeling nervous and anxious, and feeling calm throughout the day, throughout the week, but when that pattern sticks with you for a week, all of a sudden now we have a characterization of a disorder. It's really the length of time experiencing these things.
Now the question is, why 2 weeks? And that, I'm looking at the clock because I can't believe that's right. Are we supposed to end at 5? Is that right? I better get to my main message, which I haven't gotten to yet. But I'm going to start to right now by addressing this question. I've hinted at it a little bit, but why 2 weeks? There's nothing magical about 2 weeks. There's nothing inherently correct about 2 weeks. It's just something that we agree on. Right? And that's why we need to operationalize our study of psychiatric disorders. And that's why probably all of this evidence leads us to a place where we believe that psychiatric illness is real, it's biologically based, it's brain based, but the way we're defining them is not necessarily valid. And what I mean by that is the difference between reliability and validity. Reliability is Dr. Peltz and another well-trained psychologist. They see the same individual, they evaluate that same individual, and they both come to the same diagnosis. Psychiatric disorders are very reliably diagnosed by well-trained clinicians. No problem. They just don't represent the true underlying state of nature. The DSM does not carve nature at its joints. It carves nature up artificially based on a medicalization model that allows people to get reimbursed for treating something. Because if somebody's kind of sick, how do you explain that to insurance? Should you get kind of paid for that? So the problem is we exist in a capitalistic society, okay? That's truly the problem. I mean, do you see it any other way? It's a way to allow people to get reimbursed for something. Why does ADHD rely on you meeting 6 criteria out of 9 instead of 5? Surely somebody who meets 5 out of those 9 criteria is somewhat impaired in their attention or their hyperactivity or their impulsivity. But they don't meet criteria for diagnosis. They don't meet criteria for treatment. Yes?
Do you think that specifically for ADHD, they pick the number of check the boxes because of diagnosing from a medication that you use, and I also think that's like more American focused because other countries are nothing like that, and unlike every other country, America focuses on just giving meds and like finds the easy way out with money rather than finding out the real issue, because they care more about what's happening in our own world.
Is there an or after that? I mean, I guess I agree with that. I don't think there's another alternative to that. But I think, I don't want to pull the rug out from under psychiatry, nosology, which is the kind of science behind. Nosology is a very careful science. The problem is we don't have the X-ray. We don't know the biology to characterize the psychiatric sequelae. We only have the subjective reporting of people's distress. And that's pretty hard to pin down. And so, you have to agree on some number of criteria. So a lot of very well-trained, very well-intentioned people have said, well, this level of impairment is something we can agree on, is kind of meeting the standard where 95% of people who have this number of symptoms are functionally impaired. And what I mean by functional impairment is they don't like what they're feeling. They're not being successful in school, job, relationships. They're not able to take care of themselves well, et cetera, et cetera. So, you have to operationalize it somehow, but it doesn't mean that we're necessarily kind of treating it as it exists in nature. As it exists in nature is continuously distributed. It's not discrete, right? But in order to say this person's eligible for treatment, we have to say they've crossed a level. Where do you put that level? People could disagree. They do. We change the criteria. But you have to make some distinction in order to initiate diagnosis and then treatment. So it is arbitrary. But how these things really exist in nature actually is on a continuum. It's all dimensional. Anxiety is not present or absent. It's dimensional. There's degrees of it. Depression is not present or absent. It has to be for the purposes of diagnosing. It has to be in order to initiate treatment. But that's not how it exists in nature. So that's the fundamental premise of RDoC. The research domain criteria was an initiative that was kind of cooked up by the best minds in science and clinical treatment to say the way we're going to continue to decide who gets diagnosed and treated accords to the DSM model, criterion-based, discrete, yes or no. But we need to do science in a different way. The science has to understand that those diagnostic labels mean nothing, actually, and that people with a diagnosis of schizophrenia can also feel depressed. People with bipolar disorder can also experience psychosis, and so on and so forth. So the RDoC approach is more about the dimensionality of psychiatric symptomatology, not about diagnoses at all. So that's kind of where we have to probably end, although maybe I need to schedule another lecture or something, but yeah.
Come out. We'll actually, you can be the in-house. We'd love to have you here, but do you actually have a slide that might show where some of the RDoC criteria are to make it a little bit?
Yeah, that's a great idea. Let me see what I could pull up. Think of it this way, okay? Let's do this slide. This is the DSM paradigm. I'm not going to go full screen, but you guys can see that, okay? So the DSM paradigm, remember the fuzzy border that I drew around my own kind of phenotypes? That's the same here. DSM disorders are phenotypes that you could study in a genetic paradigm, in a neuroimaging paradigm, et cetera. DSM disorders are dynamic. They change, okay? You might get referred for depression, but then you might start to show anxiety. Your attentional problems might get worse or better. The diagnosis, you meeting criteria for the diagnosis is labile. It is dynamic. It can go away with treatment, right? It's not a fixed trait in you. And at the opposite end of that, kind of we're using these very arbitrarily defined things to try and do science about them, which is tricky. And we're going backward from the diagnosis to gene expression, from the diagnosis to protein expression, from the diagnosis to neuroimaging results, or neuropsychological functioning, or symptoms, okay? And then consequently back to the origin, the genes and the environment of things. And the RDoC approach says that's fundamentally wrong. And I've shown and summarized a lot of evidence here that says that that's probably wrong. But the RDoC approach flips that on its head and says, let's start by looking amongst people who have a certain level of psychological distress and functional impairment. In other words, they don't like what's going on in their brains, and it's impairing their ability to be the person they want to be. Regardless of what diagnosis they have, let's study these people and find commonalities among some of these people in terms of their genes, their environment, their gene expression, protein expression, brain imaging patterns of structure and function and blood flow, their neuropsychological function, etc. And then maybe at the end of it all, we'll come up with a better taxonomy for psychiatric illness that is embedded, and in fact, not just embedded in, but arises from our understanding of the aberrant biology rather than the opposite, right? Starting with these amorphous diagnoses and trying to figure out their biology is 100% bound to fail. Finding people who collectively have psychological distress and functional impairment and finding groups among them who have commonalities at the biological level is probably a little bit more likely to succeed. So that's the RDoC approach in a nutshell. How's that work? Does that work? Okay. All right, cool. Well, I prattled on and on, but I said the objective was to have conversations. I hope that some of these questions have been illuminating and provocative for you guys. I am actually happy to maybe do another lecture on Zoom or something now that we're all acquainted or something, and we could finish the conversation about what I study within RDoC, which is the reward system, okay? And the way I've studied the reward system is not by ascertaining kids with ADHD, which is a dopamine system disorder. The main mode of treatment is through dopamine transmission blockade. I don't study kids with ADHD and then try and figure out like what's the reward processes that are aberrant in kids with ADHD. Or depression. Depression is marked by kind of reward insufficiency. I don't study people with depression. What I study are people who are developing typically without any kind of impairing psychological distress, and people who have psychological distress and functional disability, and then try and find differences between these groups as well as kind of continuous changes across that range of abnormalities or changes in reward behavior that correlate with certain sets of symptoms, regardless of what. In the process of doing this, I end up studying a lot of kids with ADHD. I end up studying a lot of kids with anxiety, adults with depression, et cetera, et cetera. But that's just a consequence of me ascertaining a broad range of people and trying to figure out the biology underlying reward behavior, right? And then maybe you find some kind of reward syndromes that have biological subtypes under them. So I'd be happy to come or virtually come and do another lecture on that topic as well. But I hope I've kind of impressed upon you guys more than anything else that psychiatry is in need of a lot of work. It's in need of a lot of hardworking people. When I don't think there was much a part of me that went into psychology for the reason that it would never be finished and I'd always have something to do. But I remember thinking like, I don't want to go study cancer or HIV or heart disease. I think those are tractable problems that other people can work on. But I went into it because I thought it was the most complicated thing that I could work on is the brain. And so I think for those of you considering going into psychology, which you all are, I think you're going to have a lot of work to do, but I think that your orientation toward it needs to be grounded in the reality that you're not in orthopedics. We're not dealing with x-rays. We're working toward that. You guys could work toward finding the x-ray or the blueprint of certain biotypes in psychiatry, and there's a lot of value in that. But there's immense need for hardworking people who are interested in these questions. And so I'll leave it at that.
Want to say that if there are any specific questions for Dr. Glatt, we can take a minute.
Yeah, of course, absolutely.
And then also, if you want to speak to more about SUNY Upstate or about any more specific work
Absolutely, yeah. The surf program is wonderful. Brendan, is that right? Brendan can maybe speak to that. I mean, you come to Upstate, there's a stipend, you get to be in a lab for the summer and do hands-on research, which you may or may not have gotten exposure to. Our surf program is wonderful, and it provides people an inside track to get to graduate school too. I've been a mentor in the surf program in the past. I haven't recently, but maybe I will. But I've trained my share of graduate students. I've trained 5 PhD students who've gotten their doctorates and moved on to other things, a bunch of master's students and so on. But there's a lot of great PIs at Upstate, and I think neuroscience, psychology, like brain sciences is one of the premier strengths, if not the strongest thing we do at Upstate. We've got strengths in certain things. We're not Harvard. We can't do everything impeccably, but we do brain sciences pretty well. So if you're interested in the brain, if you're interested in graduate school, particularly for like neuroscience research or experimental psychology, hope you give Upstate a try. We don't have a clinical psychology program, but I've also had 2 clinical psychology PhD interns from Syracuse University, which is just across the street, do their research on my RDoc project. So we have data that I've collected with my team at Upstate from over 2,800 people in this RDoc paradigm. We ascertained, maybe I'll tell you this next time, but we've ascertained based on a kid between the ages of 6 and 12 with any psychiatric diagnosis, any impairing psychiatric diagnosis, and when we ascertained an equal number of kids who are typically developing with no concerns. And then we ascertained their siblings and their parents. And so we got about 900 parents, about 3.1 people per family. So it's a family study, so we can look at the familiarity of reward behavior. We've collected genetic data from these individuals. We've got some peripheral blood gene expression data. One of my MD PhD students used our brain genie algorithm from the blood-based gene expression to impute what's going on in people's brains and how that relates to reward behavior. So there's a lot of cool stuff you can do. We don't have a clinical psychology program, but clinical psychology interns at SU come and have done published papers in my lab on these data and so on. So we have this massive database of 2,800 people and all sorts of laboratory tests that they've done, kids as young as 6. And then you can look at kind of the biological basis of reward behavior, which is really interesting. And you see a lot of interesting things from some of these psychological tests, like discounting tasks. Like if I were to ask you, I mean, would you like, if you had to choose between $1 today or $5 in a week, which would you choose?
Probably have to say the $5, but that I want the $1, but I'm trying something new.
Oh, okay. See, this is a validity check. Now she's undermined our algorithm, but we'd ask her that probably 100 times in different ways, and we'd pinpoint what her true leaning is, right? Right? You want the $1. So, okay, well, what if I said, now based on your first answer, that you might take the $1? Okay, what if it's $1 now or $10 in a week? Isn't that interesting? Now, I bet there's some people here who instantly say, I'm waiting, right?
I feel like I'm willing to wait.
Okay, you're willing to. But there's some people who even at $5 like, of course I'm gonna wait, right? So everybody's different, but our premise was that things like that run in families. There's genes that relate to that, and that relates to different dimensions of psychopathology. What if I asked you, would you take like a 100% chance that you're gonna have $1 or 50% chance that you'll get $2? 100%. You want the sure thing. You're not a risk taker. Over the long run, over 100 trials, if I gave you $1 every time guaranteed, or you had a 50% chance of $2, you'd end up with the same amount of money at the end. But you want the sure thing, right? What about you? Yeah. Okay. There's some people who would say, yeah, I'll take a chance, right? Right? So everybody's different. So there's these discounting tasks, and we have people like do different tasks on a keyboard where they're willing to work more or less for certain rewards. We pay people actual money. Kids, this is a huge motivator. But even for adults, we give them money to do these tasks. And so we've done a lot of laboratory-based tasks on the computer, so it is operationalized, it's not subjective, but we've also had them report on their psychopathology, and then we look at the relationships within families and across people between genes, reward behavior, and psychiatric symptomatology. So there's lots you could do in research. I hope you guys consider Upstate for your career and educational needs, but consider our surf program. Certainly it's going to help you no matter what you decide to do after you graduate. All right. Thank you guys.